will talk today about progesterone receptor isoforms in breast cancer. I have no disclosures. And as this is a breast cancer meeting, I will not tell you about the relevance of studying breast cancer. I will start directly telling you that 75% of the breast cancers are of the luminal type, and they express estrogen and progesterone receptors. The current endocrine therapy that is available now targets ER, that means tamoxifen, aromatase inhibitors, fulvestrant, together with P3K mTOR inhibitors or CDK4, CDK6 inhibitors. Progesterone receptors, on the other hand, are only measured routinely as surrogate markers of estrogen receptor integrity. However, we all know that progesterone receptors and estrogen receptors interact one with each other. There are many papers in the last years. So we can think if we have a cell that is exposed to progesterone, for example, progesterone will activate all these signaling pathways, MAP kinases, P3K, STATs, that will ultimately also activate progesterone receptor and estrogen receptor as well, forming these complexes here at the promoter of target genes such as cyclin D1 or MYC. So in this context, it's very easily to think that all therapies that will target estrogen receptor will also affect indirectly progesterone receptor, and vice versa, all therapies that will target progesterone receptor will also affect estrogen receptors. What about progesterone receptors? Well, we have two main isoforms, PRB and PRA, that are both coded from the same gene, so that PRA is included in PRB. It's a truncated version of PRB, so it lacks the first 164 amino acids here. So it's very difficult to develop antibodies that will bind PRA without binding PRB. And this may be one of the reasons that research in PR isoforms in breast cancer has been a little bit delayed. Why we are interested in progesterone receptor? Well, this is so because many years ago, we developed an experimental model in which progestins induced mammary carcinomas in bulb C mice. And these tumors were hormone dependent. They need the hormone to grow. And at that time, the current dogma was that estrogens were the stimulatory hormones and progestins were the inhibitory hormones. But after the reports of the Women Health Initiative study in 2002 and the Million Women study in 2003, that they showed that women that were under hormone replacement therapy treated with MPA and estradiol, they had an increased breast cancer risk. Then scientists started to think in progestins, maybe they might be also inducing proliferation of breast cancer. And this is a controversial issue nowadays. The beauty of this model that we developed a long time ago is that these tumors that originally were hormone dependent, they needed the hormone to grow, they evolved to different stages of hormone independency in which they don't need the hormone to grow. But even though they might respond to an endocrine treatment, in this case with the endocrine treatment is mifepristone, that is an antiprogestin. And these tumors show higher levels of progesterone receptor A than progesterone receptor B. This is a Western blot in which we can see here the different staining intensity, and you can see here that this band is more intense than this one. So if we quantify this, the ratio of PRA will be higher than PRB, you will have a high ratio. So we call these tumors PRA high, and I will talk about this during my talk, so please remember PRA high means higher levels of progesterone receptor isoform A than isoform B. And how did this, sorry, something happened here, I have to go backwards. Okay. There were some slides that are missed here. Well, nevertheless.
No problem.
It's okay. These tumors regress with antiprogestin treatment. As you can see here, this is a slide that is missing in big. in which I can show here different antiprogestins. You can see here, for example, aglepristone is able to induce almost complete tumor regression. And mifepristone, on the other hand, I'm going to remove this because it bothers there. So, and this mifepristone induces a significant inhibition of tumor growth, but not as strong as aglepristone. And telapristone acetate is another novel antiprogestin, also is able to induce regression or tumor inhibition, but not as strong as the other two antiprogestins. So, but if we increase the doses of these antiprogestins, we will have the same effect. But what happens if we stop the treatment here and we leave the animals alive for a long time? You can see here in these curves that aglepristone treated mice showed no recurrence, and very low levels of recurrence was observed in mifepristone treated mice. But the difference was that mifepristone treated mice, we have to remove the tumors by surgery first, because if not, they would have grown because they had already a tumor there. But if we remove the tumor, we will find no tumor recurrence, even after 600 days, that for a mouse is a lot. It's almost, they lived 2 years. This was an adjuvant, a neoadjuvant setting. And what about an adjuvant setting in which we first inoculate the tumor here, we let them grow, and then we remove it by surgery, and then we treat it with these different antiprogestins. And we follow the animals for a few months. You can see here that again, we found no recurrences with aglepristone, and we have a delayed appearances of tumor recurrences with the mifepristone treated mice. But look here, you can see for images from the lungs, and you can see here that only very few metastases with very few number of cells were detected in the mifepristone treated mice, whereas in the control mice, you have a high number of metastases and foci, and with a higher size, as you can see here in these two graphs. And this kind of tumor regression that we see with antiprogestins is really target specific, because if we transplant these tumors in progesterone receptor knockout mice, we have the same effect. And if we transplant them in immunosuppressed mice, we can also have the same effect. So this direct and rapid and prompt effect is a direct effect mediated targeting progesterone receptor isoform A. And what about the other tumors? I told you that in these tumors, some variants were responsive and others were resistant to the antiprogestin treatment. When we evaluate the progesterone receptor isoform, we can see that in these cases, there is a switch in this ratio. You can see here the responsive tumors that had higher levels of PRA than PRB. Now the constitutive resistant tumors have higher levels of PRB than PRA. So we will call this pattern as PRB high. So constitutive resistant tumors are PRB high. So as a proof of principle, we forced these responsive tumors with continuous treatment with mifepristone to develop acquired resistant tumors. And again, these acquired resistant tumors change the pattern, so now they have higher levels of PRB than PRA. So in this model, antiprogestin responsive tumors are PRA high, and antiprogestin resistant tumors are PRB high. So what is the mechanism which explains this different behavior? Well, we know that if we make primary cultures of the PRA high tumors, progestins such as MPA will increase cell proliferation and will activate the progesterone receptor on one hand, and they will recruit co-activators such as AIB1 to the promoter of target genes such as cyclin D1 and MYC. On the other hand, the antiprogestin mifepristone will inhibit cell proliferation and will recruit co-repressors such as SMRT. Here I illustrate here with yellow spots, you can see that MPA increases the colocalization between activated PR and AIB1, and mifepristone treated cells increase the yellow spots here showing colocalization of SMRT with PR. And in the PRB high cultures, the opposite thing happens. Progestins now are inhibitory and they are able to recruit the SMRT, and antiprogestin mifepristone increases cell proliferation, inducing the activation of AIB1 or recruitment of AIB1. So there is a balance between co-repressors and coactivators that will determine gene transcription and cell proliferation. Now we wanted to validate this data that we obtained in our model. We used human cell breast cancer cell lines, and for this purpose, we selected the T47D cells that express both levels of both isoforms, PRB and PRA, but we also used a variant that has been developed by the Horwitz lab in which T47D-YA cells only express PRA and T47D-YB express only PRB. So when we inoculate these tumors on one flank of the animal, one cell line, and then the other flank of the animal, the other cell line, and we treat the tumors, the animals with mifepristone, only those that have high levels of PRA are those that will shrink, and the others will continue growing. So you can see here this different pattern of growth. Then we tested in another cell line. We used the IBH6 cell line that was developed in our institute by the Lutis lab. And again, when we transfect PRA, we have higher levels of PRA. Now mifepristone is inhibitory. But when we transfect PRB, now mifepristone becomes stimulatory. But these two cell lines that I mentioned are non-metastatic, so we want to look what happened with the MDA-MB231 cells. These are triple negative cells. They don't express neither ER nor PR. But we used a variant that was developed by the Lombez lab at the University of Paris, in which they transfected constitutively a cassette in which you can express PRA if you treat the cells with ERA cell 1 because they have this inducible cassette. So when we inoculated these cells in the animals, in the non-skip gamma mice, and we treated them with mifepristone, we found that all the animals developed metastases because this is a metastatic cell line. But mifepristone or enaglipriston reduced the number of mice that had metastases, and even though the metastases were of smaller area than in control tumors. So in summary, mifepristone and enaglipriston are able to inhibit not only tumor growth, as I showed you in the previous slide, but also they are able to inhibit metastases. And then here I show you the data of a PRB cell. These cells expressing, in this case, they were expressing constitutively PRB, and all animals developed metastases, but the foci were larger in mifepristone treated mice. So you can see that these two, they are the same cells with only PRA and only PRB. In this case, mifepristone becomes inhibitory. In this case, mifepristone becomes stimulatory. So PRA levels higher than PRB means that tumors are inhibited, both tumors and metastases are inhibited with antiprogestin treatment, and PRB higher than PRA, tumors may be stimulated by antiprogestins. We wanted to know then, we asked ourselves, can we test the effects of these antiprogestins in breast cancer samples and correlate the response ex vivo with the PRA-PRB ratio? For that purpose, we had a cohort of 369 breast cancer samples that were obtained a fresh piece of tissue to make cultures. Another piece of tissue was frozen to make Western blots. And as you can see here, most of the cases had tumors of the PRA high type, so they had higher levels of PRA than PRB. And this is in agreement with the literature that is in the field, in which there are a few studies, but in all of them coincide that PRA-I is a prevailing isoform. We try to make different methodologies to culture these tumors, and the one that worked best was the one that is called tissue culture. It's a methodology that was adapted from neurobiologists, and they cut slices in a vibratome, they culture them for 48 hours with the substances, in our case it's mifepristone, and then the pieces are fixed first formalin fixed and then embedded in paraffin to perform routine immunohistochemical assays, in our case, to evaluate Ki67 expression. Here I show you an example of a PRA high tumor and a PRB high tumor, where you can see here in the HIE, the integrity of the cells. And here you can see in this case, in this patient, Ki67 was reduced as compared to the control slices. And in this case, there was an increase in the expression in this patient. And when we compare all the cases, in all the PRA high tumors tested, in all cases, mifepristone inhibited Ki67 expression. But on the other hand, in the PRB high cases, in some cases we have a stimulatory effect. In other cases, we did have an inhibitory effect as well. And in others, we didn't have any change. So PRA samples are those that are inhibited by mifepristone, 10 nanomolar, and variable results were obtained in the PRB high samples. Well, we know that actually tumors are classified as luminal A or luminal B, or according to immunohistochemistry. This is not a very exact way, but it's the way that is done routinely. Or the molecular platforms are the ones that really define which tumors are luminal A and luminal B. So we selected, this was a collaboration with Charles Perou's laboratory, and we selected 16 samples of PRA high and PRB tumors were studied by RNA-seq, and we found that those genes that were upregulated in the PRA high tumors matched with a luminal A, and those that were upregulated in the PRB high tumors matched with the luminal Bs. So there was a correlation between PRA high and luminal A and PRB high and luminal B. And the survival analysis revealed that the PRB high cases were associated with a shorter relapse-free survival as compared to the PRA high counterparts. In other words, this means that PRB high tumors are of worse prognosis than PRA high tumors. And what about the clinical parameters? We know that histological grade, the tumor size, Ki67 expression, and HER2 are all biomarkers of worse prognosis. And we can see here in this slide that a higher number of PRB high patients were of histological grade 3, higher, they had tumor with sizes higher than 2 centimeters. They had Ki67 values higher than 14%, and there were more cases with expression of HER2. So the clinical parameters here correlate, also say that PRB high tumors have worse prognosis than PRA. But on the other hand, we know that high levels of PR are of good prognosis, and in this case, PRA high tumors are the ones that have higher levels of PR. So these clinical studies are in agreement with the molecular studies that I have shown you previously. This is a controversial issue because there is a seminal study from the Horwitz and Fuqua's lab many years ago, where they proposed that breast cancer patients with PR receptor PRA-H have poor disease-free survival rates. There is another study from the Steffi Österreich group in which they show that PR methylation that indirectly means a PRB high phenotype correlates with worse outcomes. So this should be also, we should continue studying this to see really what is going on. There are several ongoing trials using antiprogestins or progestins in breast cancer with different mifepristone, onapristone, telapristone acetate, with ulipristal acetate in different settings. But the one that we are developing in our laboratory that is called MIPRA is the only study that is evaluating mifepristone in breast cancer patients selected by their PRA isoform ratio. The study, we have already finished recruiting. We are analyzing the data. This is a window of opportunity trial that is testing mifepristone given for two weeks in between biopsy and surgery. And we compare the primary outcome is to compare the Ki67 expression prior and after treatment. And we have already this data that we hopefully will present at the Breast Cancer San Antonio meeting this year. This is a collaboration that we make with the Hospital of General Pacheco here in Buenos Aires with Dr. Hugo Vaz. The criteria to select the patients, they had to have higher levels of the PRA and ratio, PRA-PRB ratio higher than 1.5, and they had to have higher levels of higher levels than 50% of PR by routine immunohistochemistry. And 20 patients were recruited. But if mifepristone will be administered in the future in an adjuvant setting, we should first test that the metastatic foci retain the same PR isoform ratio as the primary tumor, because if the metastasis, there is a change in the PR isoforms, then mifepristone will induce cell proliferation instead of cell inhibition. So to investigate this, we selected 48 pairs of primary tumors, and they matched lymph node metastasis, and we found that all the tumors that were PRA high had PRA high metastasis. So there was a concordance between primary tumors and metastasis. And interestingly, we could observe that the metastasis showed higher levels than the tumors. So that means that the treatment, potentially the treatment of these patients after tumor removal, will be probably effective because the metastases show higher levels than the tumors. The opposite occurs with the PRB that tend to lose overall receptors, PR receptors in the lymph nodes. And in the last two slides, I'm going to tell you about that there are not only two isoforms of progesterone receptor. Many years ago, the Horwitz lab have demonstrated or have discovered many PR isoforms that are induced by alternative splicing. But they were not detected as proteins, so probably they're interested in this, the study of these isoforms decrease with the time. But I will focus in this isoform PR delta 4 that lacks, as you can see here, this is the entire PRB isoform. The PR delta 4 isoform lacks the exon 4, so it has an impaired DNA binding domain because it lacks this little bit here of the DNA binding domain. It lacks the hinge region, and it lacks a little bit of the ligand binding domain too. So it is not able to bind ligands, and it is not able to bind DNA. But using an approach in which we made a pull down and further mass spec studies of these proteins that are bound to these transcriptional complexes of the enhancer of the MYC promoter and in the proximal promoter of MYC, we studied these proteins that were forming these complexes there, and we found that PRB delta 4 was there, together with PRA, PRB, and estrogen receptors. And why we were able to find this isoform that nobody could find it previously as a protein. And this was because we cultured the cells together with FGF2 instead of a ligand, of a steroid ligand, instead of progestin or estradiol. And FGF2 is a growth factor that we know that is able to activate in a ligand-independent way hormone receptors. So these activated receptors in a hormone independent way are able to activate gene expression. So it is interesting to think in this isoform that might be related to tumor progression, because it may be not recognized by antagonists. So we selected some samples of our cohort of patients in which we could find an intermediate band between PRB and PRA that matched with the molecular weight of this PRB delta 4. So we selected these samples and samples that didn't have this intermediate band, and we measured the RNA messenger by real-time PCR, and we found that these samples that have the band, the intermediate band, are those that have high levels of mRNA expression, where those that lack the band, that intermediate band, have low levels. And when we interrogated the TCGA data set, we found that these tumors, these patients that have high expression of this mRNA isoform, are those that have a decreased overall survival. That means that PRB delta 4 expression might be involved in tumor progression, and this is ongoing work that we are doing in the lab. So in summary, I have shown you that murine memory carcinomas, human breast cancer xenografts, and breast cancer tissue cultures that regress with antiprogestin treatment show higher levels of PRA than PRB. On the other hand, high PRA-PRB ratio predicts better prognosis, although this is controversial. There is also a concordance between the PR isoform ratio in primary tumors and lymph node metastases, and novel PR isoforms may be participating also in tumor progression. So I hope I have convinced you that progesterone receptor isoform ratio in breast cancer matters, and maybe in the future, antiprogestins might be used with standard endocrine therapy for a group of selected breast cancer patients. And well, with that said, I think that, well, I wish to thank, of course, all the patients. These are the members of my actual laboratory, and these are the members of that were previously in my laboratory, but are working in these projects. And all the collaborations, I would like to highlight, well, Charles Peru with the genomic studies, Martin Ava with all the bioinformatics, Silvia